Well, good morning, everyone. It's nice to be here again in St. Louis and wonderful to be invited to share some of the work that's going on in our laboratory at a Nationwide Children's Hospital. So today I'll be talking just briefly about Clin-MSI, which is a machine learning-based approach to MSI detection that we developed specifically to focus on women with endometrial cancer diagnoses. So just a conflict of interest disclosure. I have conflicts of interest, but they don't apply to what I'll be talking about today. So just a little bit of background here on Lynch syndrome genes and cancer susceptibility, probably not needed for this audience, but if you'll bear with me. This is obviously a germline mismatch repair defect that involves alterations in a number of genes listed here, leading to cancer susceptibility in multiple histologies. And again, today we'll just be focusing on women with endometrial cancer. The cancers that occur in the setting of Lynch syndrome exhibit a mismatch repair defect related microsatellite instability, often referred to as MSI or MSI high, and a corresponding elevated mutation rate. And as we know right now, based on a lot of research that's been done, these genomic features then lead to highly immunogenic phenotype, including abundant immune cell infiltration, and this is largely thought to be due to the presentation of tumor-unique neopeptides or neoantigens. Some of the background that led to the study I'll tell you about today really came out of the TCGA endometrial cancer study that I was very pleased to be asked to lead with Doug Levine back in the day. And in fact, we convened the first meeting to begin analyzing data right here in St. Louis from this project. One of the surprising findings coming out of this study was the presence of defects in polymerase epsilon. And as you can see from the Kaplan-Meier analysis, this is typically indicative of good prognosis for these ultra-mutated cancers, so-called cluster 1, and the light blue line at the top of the Kaplan-Meier curve that's shown here from the original TCGA paper in Nature. So both polymerase epsilon and MSI hypermutated endometrial cancers are thought to be potentially available to benefit from immune checkpoint blockade inhibitor therapy treatment. And again, in particular, we know that this is a hallmark of likely response, although certainly not the only one, as the biomarkers to predict checkpoint blockade response remain elusive, I would say, to this date. But an elevated mutation rate is really what we're looking for here, and the likely predictor in many cases of differential response to these drugs. So just to focus in now after that brief introduction, we wanted to develop an assay of endometrial cancers in response to some R01 funding that we received from the National Cancer Institute. And we developed this around an existing network within Ohio that had originally been designed to identify patients with colorectal cancers and to evaluate their tumors for a variety of different biomarkers, leading to differential treatment. And so we opted into this network of hospitals across the state of Ohio with our Ohio Prevention and Treatment of Endometrial Cancer study, again funded by the National Cancer Institute. So the idea behind this was to really enable testing for women with endometrial cancer diagnoses at no cost to them. The cost of the testing was supported by the grant monies, and to effectively identify those women with Lynch syndrome, also potentially with polymerase epsilon, and to provide that information back as a signed out clinical result to their providers for additional consideration around therapeutic intervention, but also obviously leading to, in certain settings, further genetic testing of family members. And so the assay that we devised within Nationwide Children's Hospital and with input from our collaborator Paul Goodfellow at Ohio State University involves essentially next generation sequencing, and I'll describe that in detail, analysis, and I'll really describe that in detail, and then clinical interpretation and sign out of these results. We knew that from the beginning we would be looking at samples that were coming from formalin-fixed paraffin-embedded tissue, and we assumed that that would be the case. We designed an assay that included a custom capture probes that targeted the 4 MMR genes listed here, plus polymerase epsilon, as well as 21 widely utilized loci for evaluating microsatellite instability. We aimed at a target coverage of over 500-fold so that we would have high sensitivity to detect these variants, and this is, again, tumor-only testing. And we wanted to be able to identify pathogenic variants so that we could have a clinical sign-out of those in the Lynch syndrome and POL-E genes with return of results. But we also wanted to really essentially develop a novel approach to using these MSI loci that were being sequenced to develop a new way of identifying microsatellite instability and using that as part of the clinical sign-out. And so to do this, we invoked a machine learning-based analytic pipeline that I'll tell you about today. One side note is that for those of you who are deep in the weeds on technology like I am, you'll know immediately that a 5-gene panel isn't going to get you anywhere in terms of efficiency with hybrid capture. So for that reason and multiple other reasons, really to enhance basic research around endometrial cancer, we added additional 72 genes. These genes were genes that were identified through the TCGA endometrial cancer studies, so we knew that they had some profile of recurrent mutations within the setting of endometrial cancer. And we also really wanted to be able to detect genome-wide copy number aberrations that might also be drivers of this disease. As such, we incorporated within the panel of capture probes additional probes for these 72 genes, as well as a reagent that's off the shelf available from IDT that essentially tiles through the chromosomes of the human genome at about 30 kb resolution to provide relatively high-resolution copy number profiles as well. So just to be clear, these last items are not being reported out clinically. They're just sort of along for the ride. And since we had all of those data, we can now and are going back to reevaluate more information from what I'll show you at the end turns out to be a relatively large number of samples. So this is just a brief table for those of you who might understand and appreciate microsatellite loci of the different loci that were included in the capture probe set and that are contributing to the machine learning analytics that I'll talk about in just a minute. And in particular, bracketed at the top are a set of microsatellite loci that are evaluated through the Promega-MSI test. You'll see this is one of the clinical gold standards that we're going to be comparing to in the course of developing this machine learning predictor. Just a brief look at how we're using the NGS data to profile the microsatellite repeats is shown here. As you can appreciate, because we're coming out of a random whole genome library, we're going to be getting a variety of capture products from the hybridization to these probes and then aligning them back around the microsatellite repeats to really appreciate where the microsatellite instability might be detected from. So this just represents the alignment. And what we basically did with these alignments then coming out of the BAM files was to use PySam to sort of evaluate the loci in isolation and to count effectively and make a table for each one of the number of repeats that were being detected. And this is just a representative example of what one might appreciate coming out of this type of PySam output. The output here then was utilized for a set of training data that came from 15 microsatellite instable and 15 microsatellite stable cases as judged by the Promega-MSI assay through a machine learning predictor, which is XGBoost. And here what I'm trying to illustrate is that we used essentially two different approaches. One was a normalized repeat length distribution from the PySam overlaps into a z-score normalized distribution, or as an alternative into an approach that calculated the mean and standard deviation for each locus as a means of reducing features for the input into the machine learning algorithm. So here we're using a gradient boosted trees approach. Please don't ask me what that means, but that's what we used through this approach. And then the model, once developed, was tested utilizing 109 other endometrial cancer samples that, again, had been previously evaluated by two so-called gold standards, so the Clin-MSI assay and also clinical immunohistochemistry to detect microsatellite instability. We also did another comparison to a currently used approach, a bioinformatic approach that is there to detect microsatellite instability. This is MSI Sensor Pro. So what I'm illustrating here are box and whiskers plots where the red and green dotted lines indicate the 95% confidence intervals for true positive and false positives, and really comparing the results from MSI Sensor Pro to those of Clin-MSI. So MSI Sensor Pro is on the top two A and B box and whiskers plots, and by comparison, the same samples through Clin-MSI, either using Z normalization or standard deviation-based normalization approaches. What I just want to quickly draw your attention to here is that there are a couple of red dots indicated for each of the box and whiskers plots. These are in comparison to the immunohistochemistry result, discordant results with respect to the predictors. And what I think you can appreciate from here and also from this table to sort of sum up what we identified is that Clin-MSI in either mode does a very good job with respect to these categorizations as stable or unstable compared to MSI Sensor Pro. Basically the same results for recall, especially when we use an expanded number of sites to feed into the MSI Sensor Pro algorithm, but a little drop in precision, and in both recall and precision when we only focus on just the 20 sites that were from the MSI loci that I talked about earlier. The last thing I want to mention here is just how do you sort of facilitate the belief in these results and what comes out of the algorithm for the clinical directors that may be signing out cases. And I think this addresses somewhat this black box of machine learning and sort of how did you arrive at this prediction. And what I'm showing here is a protocol called SHAP that effectively evaluates and provides a color-coded map for the clinical director who's evaluating and signing out these results to really understand what drove that call for a stable versus unstable prediction. And so on your left, you can see a microsatellite stable tumor illustrated, and on the right, a microsatellite unstable tumor, where the most important features are in descending order on the y-axis, whereas the x-axis gives the sort of mean model importance of each feature group. And so I think this has really helped our clinical directors understand and appreciate what's coming out of the machine learning predictor and to sign out accordingly with other evaluation of data, of course, as usual. So just to conclude, I hope I've convinced you that training machine learning models to analyze data provides this automated and, as we've found, highly accurate way to categorize the clinical samples. We do think that it's important to also provide visualization of the data for those who are evaluating the result and signing it out. And as you can appreciate, I think these results really came from a multidisciplinary team effort, which is really what we're all about at IGM, allowing us to develop high precision and recall with data from real-world samples and deliver these high-confidence results for patient care decisions. And to date, using this approach and across the last 4 years, we've profiled 825 different patient samples and provided those back to patients and providers.